So I think we can start now. I am very happy and delighted to present this new series that we have in the EIT Health Innovators community. And this time we are organizing it together with the Clinician Engineer Hub. And I am very happy to have here with me Dr. Sasha Berdichevski. I hope I'm pronouncing it well, who is a principal scientist at Emulate. So to begin, who are we and what we are doing? The Innovators community is actually the network of innovators that is dedicated to improving quality of life, active aging, and healthcare across Europe. And we are supporting innovators towards their mission of improving health and care. And the Clinician Engineer Hub is actually the hub that brings together the clinicians and biomedical engineering fields, focuses on the challenges that doctors face in diagnosing and treating patients, and how to solve these issues with cutting-edge engineering solutions, such as the organ-on-chip technology that Dr. Sasha will present to us. And who is Dr. Sasha Berdichevski? Actually, when I read your bio, I was very impressed and very happy. Talking a bit about myself, I'm also a future scientist. I am a PhD student, so reading about your experience has been very, very nice for me. So Sasha is actually a principal scientist in European Scientific Liaison, and she has more than 10 years of experience in the development of complex systems for tissue engineering, disease modeling, and safety assessment of novel therapeutics. She has worked with microphysiological systems, hydrogels, and porous scaffolds to evaluate the performance of multiple tissues in different in vitro and in vivo settings. She holds a PhD at Techion from Israel, and she developed a novel imaging methodology to identify host response to biodegradable implants and designed an injectable microcarrier delivery system for cardiac muscle regeneration. Very interesting topic. She holds two postdocs, actually, one from University of Cambridge and another one from drug safety and metabolism team at AstraZeneca in UK. And right now, as I said, since 2020, she is the lead in European field science team at Emulate. So now I give the whole stage to Dr. Sasha Berdichevski. I would stop sharing my screen. And hand it back to you.
Thank you very much for the introduction, Leana. I just want to say and apologize that unfortunately I am not able to share my screen. So I shared the presentation with the moderators and they will be the ones sharing the screen. So unfortunately, I will ask every time to move the slides forward. A little bit of technical issues, but we will just take what we have and hopefully no other issues will happen. So first of all, I want to thank everybody for registering and investing time to hear about our technology. So thank you for joining this wonderful initiative. And I really hope that my presentation is more of a general overview and it will hit the audience, which is quite diverse and it's quite exciting. So I try to do more of an introductory presentation rather than a very technical one. So the topic is the Human Emulation System, a complete organ chip solution for next generation in vitro models. And I really like the next part, actually, how me personally and my company sees that today's science is tomorrow's medicine. So on that, yes, you can move to the next slide. Thank you. So on the agenda today, I will give an overview of Emulate as a company, as well as on organ-on-chip technology as a field. And I will touch two major studies that we performed in this year. One of these is predictive toxicology, human liver chip performance assessment. And another one is the immune cell recruitment in colon intestine chip. And I chose those because these were really important studies that hopefully will be interesting for this audience because they are as close as in vitro studies can be for human physiology. Next slide, please. So organs on chip as a field emerged from the Wyss Institute for Biologically Inspired Engineering at Harvard University from the lab of Professor Don Ingber slightly over than 10 years ago. And why there was a need for such a field from the first place. Next slide, please. Well, because simple cell-based systems and animal models fail to translate into the clinic. They're simply incapable to predict the human outcome. And there was a need for biologically complex in vitro systems that use human cells to study human diseases and test human treatments. And this is where the organ-on-chip technology can bridge the complexity as well as the simplicity of in vitro studies to study all these diseases and their treatments. Next slide, please. Now, this is to talk a little bit about Emulate as a company, but actually the main purpose for me is to show that although this field is relatively new and we as a company are even younger, we're not even 10 years old and we're relatively small, this really highlights the huge need for such technologies and how well they are adopted in the worldwide. So, for instance, our specific company is working with 21 out of 25 pharma companies globally. We have over 350 installed Zoes, which are our instruments basically. And of course, our chips are used in over 350 labs. We have 5 validated organ models and over 12 biological applications available. Again, all this within less than 10 years of this field. We have more than 75 models and applications under development with our customers. So not everything we have and we published are done internally by our internal researchers. The majority actually of adoption and driving this adoption towards the clinics is done by our customers. And we filed over 390 patents as well, providing international coverage. So this is really to highlight how rapidly this field is growing and how well it is adopted within the scientific community. Next slide, please. So what is our vision as a company? So we believe that combination of human biology and advanced technology can ignite a new era in health. And we're really focused on human health. This is our primary focus. However, we also work in the veterinary medicine space as well. Next slide, please. So I mentioned biological complexity, and this is really to show how and why organ on chips, or Emulate specifically organ on chips, can be a window into human biology. These are several examples of growing such biological complexity in vitro. So for instance, the 2 top images show our intestine and airway chips. And you can see the beautiful 3-dimensional cytoarchitecture, the polarization of these tissues, the increased complexity of incorporating multiple cells that are grown in your lab, which is of course impossible to do using human cells in a simple in vitro model. Now, on the bottom, you can see on the left-hand side, example of cancer model. Again, this is to study different types of cancer grown with multiple other cells and studying metastases and treatments. You need that increased complexity. Just by growing, cells on plastic, you will not be able to study all these mechanisms of the disease and treatment. And finally, there is a video on the right-hand side. I don't know why it's not playing. Maybe you need to press on it. But actually, these white spots were supposed to be moving, and they are immune cells that are added to the chip, and you can follow them, track their movements, and see how they interact with additional cells in the system. Next slide, please. So how do we build this biological complexity? So this relies on two major principles. One is tissue-tissue interface, so interaction between different parts of your organ or tissue. Most of these tissues, as you probably know, are in a very close proximity to blood vessels. So most of our models have this epithelial endothelial interface. However, we're not limited to that. That could be any other type of interaction that is relevant for your biology. We do use extracellular matrix proteins to bring it even closer to a biological environment for these cells, and these can be tailored for your organ of interest. You can put any types of cells on the chip. It can be from different species, different sources, healthy, diseased, or genetically modified cells. So again, one of the major principles is interaction of multiple cells in vitro. Another important principle is to have the dynamic environment. In our body, the cells are having mechanical forces, they have the blood flow, they have the stretch, they have all sorts of mechanical stimuli. And this is something that our chips are able to recapitulate, at least to some extent, by fluid flow and mechanical forces such as stretch and relaxation. Now, you will see in the next slide how our chip is built to understand a little bit better how it works and how this dynamic environment is introduced. But just to highlight here, we have two channels. In each channel, there is a flow of media. Different types of media can be flown through the system, and the cells are sitting on a membrane that is stretched. And you can change the flow rate and the stretch parameters according to your model and your scientific question. And in the next slide, I will show you how this all works. So if you can play the video. Thank you. So here you can see the two channels. One channel is slightly bigger than the other. At the bottom, the smaller one is normally representing the blood vessel, and the top one is normally representing the epithelial compartment. However, again, it doesn't have to be this setup. They're separated by this porous stretchable membrane that you can see in between, and the cells are seated on either side of the membrane. This membrane can be stretched. Again, you can tailor the stretching parameters according to your needs. For example, to mimic peristalsis of the intestine or breathing motion of the lung. And here you can see how you can flow two different types of media through the system. And this provides a really big advantage for your research, on which I will discuss a little bit more in detail in the next few slides. So this is quite a small chip. It's about AA battery size. It's flexible and transparent. Next slide, please. So when we are talking about chips, you can see on the left-hand side the list of consumables. These are the actual chips that I just described to you. We have also pods. These are the reservoirs that hold the chips, and you can put there your media, as well as collect the media after it passed through the cells. We have our instrumentation that you can see in the middle. We call the one of these instruments Zoe. And this is the square one, and this is where you put your chips and put it in the incubator to really allow these long-term culturing conditions. The other instrument is the orb. It is connected to your vacuum gas and power supply, and it sits outside of the incubator. Again, all this instrumentation is basically aimed to allow the researchers to focus on the biology and not to reinvent the wheel of having complex connections and tubing and leakages. So basically, it is really there for you to do your biological studies. We also have some software apps that are under development and will be released very soon. So this is basically like a whole package technology that you can incorporate in your labs. Next slide, please. Now, for some of the scientists in the audience, a little bit more technical points. How do you really look into the biology and assess, your disease progression, or efficacy of your drug or toxicity or anything that actually you try to investigate. So there are three major ways to do that. You can sample the effluent, so basically the media that came through the cells, and measure barrier function, release of cytokines, metabolites, anything that can be measured while being soluble. So basically anything you routinely use in your lab or want to develop from anything that is in the effluent. You can also do imaging of any kind. It can be phase contrast, confocal, live imaging, electron microscopy. So these chips are transparent, they're flexible, easy to cut. So basically you can look at the spatial temporal results when you want to see whether the effect is on the endothelial cells or in a specific cell sub category. So we heavily rely on imaging, again, because it's extremely informative. And of course, you can take the cells off the chip and run them through flow cytometer or lyse them for RNA, DNA, protein, anything you would like to do with the cells in suspension. And then, of course, do any endpoint analyses that are important for your studies. Next slide, please. So the design of this chip is kind of flexible. So the idea is that you use the same technology to grow multiple organs and to use it for multiple research areas. And these are just highlighting the major ones that we ourselves as a company and our customers are mainly using. It can be admitox, cancer, inflammation, neuroscience, microbiome studies, and infectious diseases. And obviously, when you look at all these applications, you straightforward see that there is a need to have that biological complexity. You cannot study infectious disease without having different cells interacting, without being able to put a virus or a bacteria, or in some cases, incorporate immune cells. So, of course, these give a huge advantage when you have all this biological complexity for studies such as infectious disease or cancer, for instance. Next slide, please. So our models are built in a kind of expanding way. So first, these are emulate models. I mentioned first that we have several models that we established internally. We have protocols for them. In some cases, we have biokits, which means that we also provide pre-validated cells. And these are the models that we work with as a company, and you can buy them from us as a model. Next one, please. If you can press, yes, thank you. And then you can see that much more is actually being developed by our users, our customers. You see quite bespoke applications for bladder, in different infections. We recently published on the vaginal microbiome, jejunum, bone marrow, lymphoid follicle, etc., etc. Basically, most of the tissues that you have in the body were put on our chips by somebody. Not all, but definitely most. Not everything is published. Some of the data I will show you is also not published, but you just can see how diverse our chip can be for applications and models. Next slide, please. And this just to highlight some of the publications. Again, we have over 100 publications that mention using our Emulate chip or focus on it solely. And I or my colleagues are happy to forward you the relevant publications if you do reach out to us. I will leave my details and also the details of my colleagues and the website where you can find further information, but please do apply to us directly if you want to take this further for any questions or publications or anything that would interest you. Next slide, please. Now I want to move to the predictive validity study. Why did we do this study and why it really highlights the huge need for better predictive models? Well, Between 1953 and 2013, 83 drugs out of 462, which is 18% of the drugs that were granted marketing authorization, were withdrawn because they caused drug-induced liver injury, or shortly DILI, which makes DILI as leading patient safety concern of the 21st century. And here are examples why this is really, really alarming. So in the table, you can see some of the very known drugs that reached human patients, and they caused either death or severe disability. And of course, they were withdrawn, but these toxic effects were missed by the workflow that has been established by the FDA in 1953. But what is even more alarming are the examples on the right-hand side. So these 5 examples happened just this year. So it's really prominent, it's evident that current workflows using in vitro and animal models are inadequate to predict such daily toxicity, and there is a huge need for much better predictive models to identify these toxic compounds and prevent them from reaching the clinic. Liana, shall I stop for questions, or shall we address them at the end? Please let me know what is the better.
It's up to you what you prefer. If you prefer to answer them now.
I think let's cover things, and then maybe we can go over the questions, and whichever we cannot cover here, we will follow up.
Sure.
If that's okay with everybody. Thank you. Next slide, please. So this study we call is in the predictive toxicology field, and it actually was published in a Communications Medicine journal last week after a very long and laborious work from my colleagues in the US. And this study covers the predictive validity of our model. Next slide, please. Now, I will give you some of the results and explain why and how we did this study, but I do recommend for you, either you're a clinician or a basic researcher, to read this study. It is one of the largest studies in the field, and it's quite unique because it really highlights how some models are more predictive than the others for the outcome for certain drugs. So this is really comprehensive study. I do encourage you to take a look at this paper. So the objectives were, as highlighted here, basically we wanted to qualify our liver chip, again, because we wanted to focus on drug-induced liver injury. We did rely on the guidelines published by IQ-MPS affiliate. It's a consortia of biotech and pharma and some academia affiliates that work together to improve drug safety and science that eventually reaches the human patients. Again, I will show you the publication that highlights these guidelines in the next slide. We wanted to quantify the sensitivity and the specificity. If you could go back, please, yes. To quantify the sensitivity and specificity of 27 small molecules. Again, this was part of the IQ-MPS consortia guidelines. And we compared that to 3D hepatic spheroids. Hepatic spheroids, as well as simple sandwich liver cultures, are routinely used in the drug development process. So that's why we compared to the spheroids. We wanted to estimate, of course, the economic impact of incorporating such chips in the drug development workflow. I will not talk about that, but it is extensively described in the paper. If you're from pharma company or simply interested in that, again, just please take a look at the paper. And lastly, to highlight where in the drug development process we propose to use liver chip technology to improve safety of patients. Next slide, please. So this is the publication I mentioned before, where the IQ-MPS consortium actually published the guidelines of what your system has to have in order to qualify to be incorporated in such workflows. We compared that again against historical preclinical workflows of in vitro and animal models as part of the IND submission, and historical workflow against 3D primary human hepatic spheroids. Next slide, please. Let's see how we're with time. So this is a list of the 27 small molecules that we tested. You can see that they're quite diverse. They have different mechanisms that are known to cause DILI. Some of them are less toxic than the others, and some are non-toxic. So we really wanted to cover the whole range and to test whether we can differentiate between the different types of drugs, as well as we wanted to cover the majority of the mechanisms by which these drugs cause the toxicity, again, to show that increased level of biological complexity is absolutely necessary for some of these mechanisms, and having just your hepatocytes or co-culture of hepatocytes would not be enough. Now, all drugs were tested blind, which is important, and concentration range tested up to 300 times of free human Cmax. Next slide, please. So this is specifically our liver chip. So again, if you remember the video, all our chips are designed similarly, but the biology that you put on them is different. So ECMs are different, the cells that you put are different. So for the liver, we use quad culture. You put hepatocytes at the top channel, the one that is slightly bigger, and then a combination of stellate cells, Kupffer cells, and endothelial cells, liver sinusoidal endothelial cells, or shortly LSECs, on the other side of the membrane. All these cells are present and interact with each other. And then you can, of course, test different mechanisms where all these cells can potentially be involved. Next slide, please. In terms of sizing, I don't have much time. Again, I don't want to highlight too much because it's all in the materials and methods section there. But overall, we tested 27 drugs up to 8 concentrations with at least 2 replicates, in some cases more, per concentration. And in total, it incorporated 870 chips. So you can imagine how large this study was. There was a lot of work put into this study from my colleagues in the US. We measured some of the morphological and functional readouts, as you can see in the next slides. Next, please. So this is our typical organ-on-chip study design. Again, it varies between organs, and it depends also on what endpoints you want to do, but this is quite representative for the liver at least. So first, you sometimes grow your cells, you prepare everything, then you prepare the chip, you put the cells on, you let it stabilize, connect it to flow. In some cases, again, for example, intestine also incorporates stretch. You let the culture stabilize, and then you can start your compound testing, or adding viruses or adding bacteria. So in this particular case, of course, we added one of all these 27 compounds. And as a readouts, some of them were non-terminal, such as morphology, albumin, ALT, and urea secreted in the media. And some of them were terminal, so at day 7 of the treatment, which is day 14 of the culture, we terminated the culture, and we either lysed the cells or stained them for immunofluorescence. Next slide, please. So yeah, this is not the updated presentation, but that's fine. So one of the criteria that was put by the IQ-MPS consortia was really to make sure that the cells are retaining the right morphology, the right expression of markers. And this was one of the first criteria that one needs to pass in order to qualify for using such system in these studies for predictive toxicology. You can see here some of the examples of the hepatocytes and non-parenchymal cells that have the right morphology, as well as express the markers. For instance, hepatocytes have the cuboidal morphology, have abundant mitochondrial content, polarized cytoskeleton expression. MRP2 is a bulk molecule evidence, as well as presence of Kupffer cells and stellate cells, and the CD31 showing the beautiful connection of the liver sinusoidal endothelial cells growing in the culture. Next slide, please. Here you look at the transmission electron microscopy. Again, the cells were visualized on either side of the membrane. And for the top side, you can see it in the image on the left, you see the formation of bile canaliculae. You can see it with a white arrow. And red one actually shows it's not really in place again. This is not the last version of the slides I sent, but that's okay. So you see these little dark dots that show the accumulation of albumin. Yeah, so this really shows the hepatic function. Thank you very much for highlighting that. On the right-hand side, you see the adjacent Kupffer cells and endothelial cells that are really packed close to each other. And of course, the typical elongated structure of the Kupffer cells with the pseudopodia and lamellopodia. Next slide, please. So morphologically speaking, we passed the first requirement. Now we wanted to look at the function. And two of the most important functions of hepatocytes is really producing the albumin and urea. And this is something we routinely use to check also the health of our culture, but also response to toxic compounds. You can see that in both cases for albumin and urea, the values were within the range dictated by IQ-MPS consortia. Next slide, please. And additional data here shows the RNA-seq of phase 1 and 2 enzymes. The requirement was, if I remember correctly, to have 17 of those transporters, and we had 13 of them maintained over the culture time. So the majority of these were expressed in a stable way throughout the culture. So again, it's just another level to show that our liver chip is good to be used for such a type of studies. Next slide, please. Now, this is a very important slide, and it shows example of one of the two structural analogs, and it is aimed to really show not only are we able to pick up toxic compounds, but also are we able to distinguish between more and less toxic structural analogs. So chemically, they are very similar, but some of them caused severe toxicity in clinics and some did not. And this is one of such examples for trovafloxacin and levofloxacin. And you can see that for albumin, ALT, and morphology, all the three parameters were different for less and more toxic equivalents. So basically, liver chip was able to distinguish between the two. Next slide, please. And this is just another one to show also by imaging how beautifully the chip can recognize also the mechanism of toxicity of one of these more toxic analogs, trovafloxacin. So basically its mechanism of action is through apoptosis, and we're able to pick it with caspase 3,7 cleavage in the top row, but did not pick it up for levofloxacin in the bottom row. Now, just to emphasize that the concentrations here are different because these are multiplication of human free Cmax. That's why they're not the same concentration. So we always normalize that to what is used for human Cmax. Next slide, please. And this is a summary of such 7 pairs. For all of them, we were able to distinguish between the less and more toxic equivalents. Again, just highlighting the very beautiful sensitivity of this platform to pick up toxicity properly. Next slide, please. So all this was to show that we have met the majority of the criteria sent by IQMPS, and that's why we're qualified to look how we can predict drug-induced liver injury. Now we actually go to the real data. So next part, we did look at the margin of safety of with and without protein binding. Again, we don't have much time, and I really want to cover immune cell recruitment as well as leave some time for questions. So I will not get into detail how we did and why we did that. It's all in the materials and methods in the paper, but let me really go to the results. So next slide, please. So here we did calculate the sensitivity and specificity for different scenarios, 1 donor, 2 donors, and 3 donors, compared it to spheroids as well as animal models. Now, all these animal models we did not run ourselves. This was calculated from the fact that all these 27 drugs reached clinical studies, thereby they passed in vivo studies. There were not able to pick toxicity in any of these studies. That's why the 0% sensitivity. And you can see that if we do not account for protein binding, but do use 2 donors, you can reach 80% sensitivity and 100% specificity as compared to spheroids, which are only on 42% sensitivity. So it's almost double, meaning that our system is, of course, much more sensitive than spheroids and even more sensitive than animal models. Next slide. There was supposed to be another slide. Can you go back, please? Yes, thank you. So this one highlights what happens if you incorporate if you account for protein binding, which of course is the much better approach. And then you can reach 87% sensitivity. So it's even higher when you do your design in a way that you account for protein binding. Again, it's higher also for the spheroids, but we are almost twice as sensitive as spheroids. And again, animals are, of course, still at 0%. Next slide, please. This is where we propose to incorporate liver chips or potentially other chips, other models to test for toxicity for much better prediction. And the idea is, of course, to start simple, to use and screen your compounds in traditional in vitro models is quicker, cheaper, of course, this makes sense. And then when you narrow down the number of candidates, to run them through organ chip first, because again, these are much more predictive. They use human cells. That's why the probability to get the correct result is much higher. And only if they pass to move to animal studies. And I personally, I don't speak for Emulate, but I personally believe that gradually animal models will just be used less and less and less until eventually they will cease being used at all, especially after the FDA Modernization Act came out, and also other authorities are moving away from animals because it's absolutely clear that rodents cannot pick up most of the things. But at the moment, of course, it's impossible to completely eliminate it. That's why we propose to put the chips before the animal studies if they don't show toxicity. It's just, if you can see this workflow, then of course there is another level of testing. So before it reaches human patients at clinical trials, to increase the level of the candidates and to reduce the number of toxic compounds reaching human beings. Next slide, please. This is a summary again for the sake of time. I will not go over that. I hope I was able to show you that this really tremendous effort that was put to reach these results. And again, I encourage you to read the paper. But we did show that our liver chips outperformed animal models and 3D hepatic spheroids, and we do recommend to incorporate them in routine workflows for drug development. Next slide, please. Now, a little bit about immunology and modeling immune cell recruitment in colon intestine chip. Next slide. This is one of our most important models that I personally like a lot because it is extremely complex and you can do so much using our intestinal chips. Honestly, it's really exciting to be part of that. So for this particular study, we did use a colon. However, we do have other parts of human intestine as well. Next slide, please. And this is how we grow human intestine. This is slightly different from the liver. You can see again that the biology is different while the chip is actually the same chip. So we use human-derived, patient-derived organoids. They're grown in, how you would normally grow organoids statically, but then they are broken to fragments and they're put on the chip with the relevant endothelial cells on the other side. So the same endothelial cells you would take from that area of the intestine. And we have a substantial amount of data to show that having the flow, stretch, and these supportive cells really improves the physiology, the morphology of the intestinal organoids in culture. So it is important to have all these to grow your cells. Next slide, please. And this is really a schematic to show what is immune cell recruitment for people that are not familiar with that. So it has several steps. Can you move forward? It goes in kind of sections. One more. And I think, yes, that's all. Thank you. So basically, it shows all the steps of such immune cell recruitment. You flow your immune cells through the blood vessel compartment, again, because it makes sense. That's what's happening in your body. And then having the flow helps for these cells to pass through, but also it is absolutely necessary for the cells that are attaching, only those that should attach will attach. And again, I hope I have time to show you some of the data. Then these cells pass through the endothelial barrier and the membrane into the epithelium, which is inflamed. In this particular case, we did model it for IBD, irritable bowel disease, but you can of course model it for other diseases. This was really using the chemokines that are relevant for this particular disease. And then of course having these immune cells acting on the other epithelial compartment have the effector function, and they cause for cell death and release of the cytokines, and eventually further cell death and the damage of the barrier. And this is exactly a very complex multi-step mechanism of human disease. This is what happens in IBD. And the question was, are we able to recapitulate that in vitro? Because this is extremely complex. Next slide, please. So again, this is a typical study design. I won't get into detail, but again, you first establish your culture, then you add your chemokines, your cytokines to prime the endothelium to be inflamed, as you could see in the IBD patients. This allows, of course, for immune cells also to attach and migrate into the other channel. Then at the end, again, we sample non-terminally, and then at the end we fix and image these chips or lyse them for the RNA. Next slide, please. How much time do I have? So I don't have much time, so I will maybe skip some of the data. But here is really to see whether the treatment, the inflammation of the endothelial compartment caused an increase in the attachment of total PBMCs, total immune cells. And you can see that when treated, it did cause much more significant attachment of the PBMCs to the endothelium. So also it can be quantified using microscopy, as we did in the graph on the left-hand side. Next slide, please. So again, this is video. Unfortunately, it's not working. Maybe you can try and play it. No, okay. So that's unfortunate because I find this video very exciting. So these white dots are the immune cells and the blue arrows highlight the pores. And you can see the video was working, that these immune cells can be tracked passing through the pore from the bottom of the chip into the top. And this is really dynamic live imaging, looking at these cells in motion as a reaction to the stimulus, which I find absolutely extraordinary. Next slide, please. And this is to show a cross section, so you can look at the chip from the top, as in the top image, but also you can look at the cross section either by sectioning or by confocal microscopy, and you can track how many of these PBMCs were attaching to the endothelium, stack or passing through the membrane, or passing to the other side into the epithelium. You can quantify that, and of course you can also stain and look which subcategories of the immune cells were recruited and attached. So this really gives you a deep mechanistic understanding of what is happening. Next slide, please. So this is really to show that it can be very specific in terms of the cells that are recruited. So a specific subpopulation of these PBMCs that is relevant for the IBD was recruited as compared to what was dosed. And again, you can do it by either flow cytometry or immunofluorescent analysis, as here. Now, all this data is still unpublished, so it's quite exciting to present that. Next slide, please. Now, this is very important slide because there are multiple models that test similar immune cell recruitment or immune cell attachment in static conditions or in reciprocating flow or whatnot. And this really is here to show that there needs to be a high flow or relatively high flow to make this inflammation-specific recruitment of specific subcategories, subtypes of these cells. And you can see that when the flow was low, as on the right-hand side, the same number of total PBMCs attached. However, having the high flow was much more selective for the attachment and increased it 6.5-fold as compared to the control. Yeah, for the priming stimulus again was the inflaming the endothelial cells, the blood vessel compartment. So this makes sense because you need a much stronger interaction between the immune cell and the endothelial cell for this cell to really retain, while those with much less strong attachment are just flushed. That's why this is much more selective and much more representative of human physiology. Next slide, please. So the one of the last points of such recruitment is, of course, the effector function, as I mentioned, and the release of pro-inflammatory cytokines such as IL-22 or interferon gamma. Again, you see that when you have the priming stimulus, when you inflame the culture, there is an increase in secretion of both of these important cytokines. Again, this is in correlation with what we see in the clinic for IBD patients. Next slide, please. And lastly, the barrier damage. We can measure barrier function by using apparent permeability. This is a non-destructive test that you sample your fluorescent tracer from the media. And you could see that, of course, with treatment and with over time, this barrier function was damaged. Again, this corroborates the findings from the clinic, and this was expected, and we were very pleased to see that it can be also recapitulated in vitro. Next slide, please. I think it's the last slide, and then we have a little bit of time for questions. So this is immunofluorescent imaging, really to show that what we saw through the media, the slide before, can be also tracked visually, which is of course very important. And we focused on the expression of tight junctional proteins. And you can see that both untreated control, but also the stimulus, the priming stimulus without the immune cells did not cause any damage to tight junctions of the epithelial layer. However, when you did have the stimulus and had the PBMCs running, this barrier was almost completely disrupted after 8 days in culture. And as a positive control, we ran interferon gamma, which is a very known, important effector for barrier disruption. So you can see very similar results between the positive control and our group where we use both the immune cells and the priming stimulus. So again, it's just another level of confidence that this model works properly and gives the results that you see in the clinic as well. Next slide, please. I think it should be the last slide. Summary of the last part of the immune cell recruitment. We see it as a holistic model for immune cell recruitment because it recapitulates inflammatory response, including attachment, migration, effector function, and barrier damage. Again, this is very complex, and being able to reproduce it in vitro was extremely exciting for us. It does have tissue and inflammation specific recruitment. We have more in-depth data, and I encourage you to contact us directly to get more in-depth discussion about that if you're interested in this area. This was IBD specific, but again, one could use that in other models and other applications. We do have the relevant shear for such recruitment. This is very physiological and as close as possible today to human intestine. And again, we have all the pre-qualified biology and scientific support to give you if you would like to work on something as complex as that in your labs. And I think the last one is just a thank you slide, unless I forgot something. Oh, yes, I forgot. Yeah, that's important. I really would like whoever attended these, but also your colleagues, maybe you can share with them, to join our growing organ on chip community. It is growing worldwide. It is very important that even if you don't use our technology, at least you're aware of what is happening in the field. I truly believe that this field will continue growing and expanding and being more and more complex and closer to human physiology. So you can join our MOXIE Emulate website that is highlighted here. This is a community for users or non-users that want to learn about the technology, share experiences, ask questions. Basically, it's like a dialogue. You can share images, anything of that nature that is related to organ on chips. You can register there for free. You can visit our website that has a lot of information. You can do your work yourself. If you could just reach out to me or my colleague Ben, it's not Bear, it's misspelling, is Ben Howard. And our email addresses are here, and we will be happy to address any of the questions and forward you any of the relevant information. So just please reach out to us, and I am really happy to have presented that. So thank you very much for your attention, and I think we have 5 minutes for Q&A. Liana, will you read them, or shall I read them, or you are
Thank you very much, Sasha. Actually, we just had one question from the audience, from Bea, if the Emulate system research is only for research or if it's also clinical. And your colleague actually already answered to that question. But I myself have a couple of questions. As you mentioned here, neuroscience, and I'm from neurosciences field. Could you talk a bit about what kind of chips do you have for neuroscience field and what kind of studies could be done? How long could the study go with the chip? What is the longest time point you can achieve?
This is a very good question. So if you remember one of the slides, and I don't know if you do because there were many, we do have brain as our internal model. So meaning that we do have a BioKit. Now, at the moment, we're working on qualifying the cells for the BioKit, because they do not pass our strict criteria. So we're still working on qualifying the cells to be able to provide users. But this is actually one of our most complex models. You can put multiple cell types. You have also some information on the applications on our website. But generally speaking, you can put, for instance, microglia, neurons, endothelial cells, any types of the cells in different combinations. Grow them on chip. It depends on the application, whether you are interested in blood-brain barrier, neuroinflammation, how long your exposure or disease needs to be done. And this is something we normally discuss in depth per scientific question. So if this is something you would be interested for your research, one of our scientists will have such a dialogue with you, ask what your scientific question is, whether it's disease or testing compound transition through BBB or things like that. But most of our models fall within 1 to 3 weeks of culture. Some are longer, such as airway, but it's 1 to 3 weeks normally.
Okay, that's super interesting, as I also work with animals, and hearing that there's some new technologies we can use to reduce the animal models, it's very important as well. And just as my colleague reminded me in the chat box, please take a minute and give us your feedback about this webinar, not just for us as the community, but also for Sasha to know how about her presentation. As for me, it was super interesting, and the amount of work that has been done is just tremendous. I would really like to congratulate you and the whole team that has been working on this. I see that some of your colleagues are also here to support you, which is also very nice. Unfortunately, we don't have time for much more questions, but of course, we will share your contact details so that people can contact you. I am happy that we had this webinar and we listened to you. If you have any questions for the audience, maybe, or some take-home message, we would love to hear from you.
Okay, thank you very much for organizing that. That was a pleasure to be here and to be part of Emulate and this growing organ-on-chip technology field. This is exciting time. Thank you, everybody. Enjoy the rest of your evening. I don't know in which time zone you are, rest of your day, Musab and other people on the other side of the world. Thank you.
You are not in the same place even. That's amazing.
No, I'm in the US, so yeah, thank you very much. And
thank you very much.
Your day. Take care.
From my side, just the last message is to wish everyone happy holidays, of course, in all parts of the world. And we will now have this new Talking Health Tech series. So we hope to have some new episodes soon. So if you have any ideas or if you are part of a company, we would love to hear from you. Thank you very much, everyone.
And happy holidays, everyone. Happy New Year.
Bye-bye.
Bye.
Visit community.eithealth.eu to join our community and shape the healthcare landscape with us. For more such videos, subscribe to our channel.